Good afternoon to everybody. Welcome in my office. This feels somewhat odd because this is the place where I have written most of my thesis. So now I am defending it at the same place. And today we are here to. Now I'm going to introduce you my thesis. As you know, the title is Determinants of Spatial Patterns in the Composition of Plant Communities. As ecologists, we all know that many phenomena in nature form a sort of spatial patterns. But how can we look at the spatial patterns? There is one perspective. According to it, they are a kind of nuisance, and for a good reason, because if you imagine you're sampling over some patchiness, then your samples they do not bear independent piece of information. They are spatially also correlated or pseudo-replicated. So one thing you can do with it, you can simply avoid it while sampling. You can place your samples far away from each other. Or there are other things you can do. You can do some post hoc fix. There are many methods now how you can deal with spatial dependence in your data if you are not interested in it. But there is another viewpoint which sees spatial patterns as something very interesting and useful and worth exploring, because they must be there for some reason, because of some processes that are driving the communities. So the thing you can do is to explore them, build hypotheses, test them, maybe collect new data, and repeat the whole process again and again until you arrive at some new knowledge. Now we can proceed a bit further. If you are really interested in spatial patterns, you can say about them something based on how they are originated. So if you imagine some environmental patchiness and there is some, let's say, vegetation patchiness superimposed over it, then you can say that the part that is overlapping with environment, it's caused in an ideal case by environment. It's an induced spatial dependence. And the other part that is left over, but it's still spatially also correlated, is called true spatial autocorrelation, or I will refer to it as pure space throughout this depends. And the origin of this pure spatial fraction, it's more complicated. It could be due to some dispersal or other intrinsic processes like biotic interactions. And now there is also a methodology that was designed specifically to tease apart this environmental and spatial variation, and it's called variation partitioning. And what I showed you could be in this framework aligned to this middle part, which is spatially structured. So here is the induced spatial dependence, where the spatial and environmental part overlap. And here is the pure space. And you can also have this unstructured unstructured variation, either explained by environment or completely random. And now we have this tool in place. And what can we do about it? Actually, this variation partitioning was introduced in early 90s, but it took almost 10 years until it started to be a really very popular tool in the spatial context. And it was around the debate when the neutral theory of biodiversity and biogeography was introduced. And it was also later included within the framework of meta-community ecology. According to neutral theory of biodiversity, the species do not differ in their ecological requirements, and it's because the individuals don't differ, because it's a matter of chance whether an individual will reproduce or die, and the dynamics within neutral communities, it's caused by this stochastic random drift, and it could be also caused by dispersal if it's included. And this is in stark contrast with our intuition about how the nature works, which was been covered by the traditional niche theories. So when you take this tool, variation partitioning, you can say that if a community is somehow influenced by niche processes, it should have some connection to environment. So you will get some sort of environmental variation, spatially structured or not. And if it is under neutral dynamics, there could be either this stochastic, random, unexplained part caused by drift, perhaps, and this spatially structured part, which could be caused by dispersal. So this dichotomy was included into meta-community ecology, but there were some other parts, some other theories included as well. It was inherited from meta-population theory. And here, what is interesting for us is that part of this meta-community ecology assumes heterogeneous environment. It includes these niche dynamics and also some mass effects. So if we do variation partitioning, we can also align the processes to this fraction of variation, although here it's more complicated. And part of it assumes homogeneous environment, or the environment has no effect at all. And as I said, there was a debate, and the debate went like which one of these theories is true. And of course, we can say this is true for all of them. It's much more complicated, and it's more productive to look at it like there are some important gradients. And if we move along these gradients, then the processes start to be more important, or they lose their importance. And one of these gradients is scale. And scale is also aligned with environmental heterogeneity. As we zoom out, there is more environmental heterogeneity encompassed, so we get more, we get stronger environmental variation. And if we zoom in, the environmental heterogeneity almost vanishes, and we get less individuals to deal with. And here in the picture to the left, there is also introduced some random noise or something. It's these black and white dots, and it's a distribution of species outside of the environment they should be suited to. It could be spatial or not, it's not important here. And as you can see, if we zoom in, this noise becomes more prominent. There is no environmental heterogeneity. So we can say that along this gradient of spatial scale, we go from deterministic niche processes at large scales to more stochastic, maybe perhaps neutral processes at smaller scales. And the other part of this reasoning, or other important gradient, is along dispersal. Now we are not shuffling scale. There is some environmental heterogeneity, but as dispersal rates change, this environmental heterogeneity becomes more and more prominent, and then again drops. And the spatial variation, the pure spatial variation, it goes against it, because as we start, as we can see to the right, dispersal is quite limited, and it causes small clumps, and species are not able to track the environment very closely. So environment won't explain much, but space would. Then as dispersal gets more important, or not important, but more efficient, then these spatial patches would, in an ideal case, they would completely track the heterogeneity of environment. And after that, if dispersal is really very efficient, then the species would be almost everywhere, and the environmental signal will fade out again. So we can also say that from a perspective of an organism, dispersal somehow also modifies the scale of perception or expression of environmental heterogeneity. So now I introduced you to these lofty theories, and I think they are very useful. They are great. They help us in at least thinking about the systems we are studying. But there's a problem that these theories have also often been followed uncritically. The literature is full of such examples. So my motivation with this thesis was that I wanted to go a little step beyond and try to evaluate what I am doing within these frameworks. Of course, this is very hard to achieve, so I'm not completely sure whether I achieve this, but it's not that important for now. And I was studying forest understory communities and trying to explore and explain their patterns. I focused specifically at small scale, and in the first step, I was doing mainly a general pattern description. I focused on community level and more on the environment. And in the next step, I descended to species level and focused on the unexplained part of variation, and specifically on dispersal as a source of this variation. As you already heard, I did this in 3 sites. It were these beautiful thermophilous forests in South Moravia, and we established there grids of plots 100 by 100 meters. This turned out to be a really challenging part of the whole endeavor, because if you are in the forest, there are trees, the terrain is not flat, and there are other issues. So yeah, the final design wasn't that completely regular as it should have been in the beginning, but I think it's still very good. And what we did there, we collected data on species composition and abundance, and later also seed bank samples. And one important thing, we collected a number of environmental factors, such as light, so chemistry and topography, because if you are trying to say something about the unexplained spatial variation, you should first know that you did your best to account for all possible environmental variation as well. I used quite standard methods, mostly based on linear models, and for modeling the spatial patterns, I use these distance-based more vector maps. It sounds terrible, but if you have a regular grid like me, you would get these nice decorative spatial patterns out of them. And apart from being decorative, they are also they the patterns from the broadest to the finest scale. As you can see, you can combine them to make even more complicated patterns. And yet they can also be represented, as you can see to the right, by these sine waves, where you have autocorrelation along some distance gradients. So let's go to the first paper, which was this exploratory study on spatial patterns in understory communities and the role of environment. So my thought here was that, well, we are at a small scale. So as I have said, there should be not much variation according to the theory, at least. And so there could be lots of spatial variation, which I was going after anyway. When I did the analysis, I found that, well, it depends. And it depends on which side you are interested in. And it's kind of trivial, but it doesn't matter because I did also this spatial decomposition of the patterns. And what we can see is that most of the spatial patterns, either environmental or pure spatial, they had broad to mid-scale size, how to say it. And after I accounted for the effect of environment, which is visible in these right plots as the difference in the height of bars, we can see that the environment accounted mostly for the broader scaled patterns, which is in accord with what we know from the literature. But anyway, there is still some spatial variation left. You can see how it looks if we plot it in the landscape context. And I was trying to find out whether it could be correlated still to some unmeasured environmental factor, because this might be a problem in interpretation. And when I did so, there was an interesting thing, because I supposed that I will find some correlations with indicator values at the size where there is a lot of unexplained spatial variation, but I found the strongest relationship at the site where it was at its lowest, which was kind of strange. And as these correlations were not so prominent, then I reasoned in the end that maybe these patterns could be also caused by dispersal limitation. So in the next step, in the next paper, I went back to one of these sites and looked more in detail whether the local occurrences in a species could be linked to their dispersal abilities. I downscaled a bit to a 5 by 5 meter grid, and I selected about 30 species and looked at these focal plot to neighborhood relationships, as you can see here in the picture. And it is important to realize that what I did was done in two steps. So first, I tried to explain the occurrences of species by their occurrences in neighborhood and by environment. The environment was accounted for to be more precise. So what I got was these variation partitions, these explained deviances, as you can see to the bottom. And as you can also see, there is always quite it's a fair share of neighborhood variation, the dark gray part. And when I did this, then I took this neighborhood variation, and in the next step, I correlated it or regressed it on dispersal traits. My expectation was that if a species is a poor disperser, then it would not be able to abandon or to disperse past its focal plot. So there will be a weak correlation to neighborhood. Then if it dispersed, not very much, but still at the scale of the neighborhood, then there will be the best fit. And then if the species disperse this too far, there will again be a weak a weak relationship. And when I did the analysis, I also used some PCA axis whenever I did an ordination on all traits, but there was no relationship. So we can see that these results are quite messy. If we made just some very basic categories out of the traits, as you can see to the right, there is something like a relationship that species with heavy seeds with unassisted dispersal tend to have a larger pure neighborhood variation, because perhaps they are fitting this neighborhood scale of dispersal, and the species that have some plumes or papae for anemochory, they tend to have low neighborhood variation. But otherwise, you see I hear very many relationships, so this is more of an exploration than real testing, because of course you will always get something significant out of this, but this may not mean anything. Why is this like it? And maybe it could be due to some methodological issues, or because, and this was the ecological explanation, other processes than dispersal may shape the distributional patterns of understory plants. I was specifically thinking of source limitation or establishment limitation, or some stochastic processes. So now let's go to the last paper, where I was again trying to establish a link between dispersal and some sort of spatial variation. This was the pure spatial variation from the traditional variation partitioning between environment and space. And here, my concern was that if we are doing variation partitioning, trying to explain the processes behind community patterns, we are usually doing it on adult plants. But the adult plants, they are already, it's an, I would say, many things happen since a seed arrives at a spot and before it becomes an adult plant. So obviously the patterns will be somehow, they will be, yeah, I'm sorry, not that perfect as they were originally, basically. So I was thinking, well, I should get some idea about the true dispersal in forest understory and how to do it. I was first trying to construct a seed trap, or seed traps, but I wasn't successful at it. And then I thought, well, seed bank is a sort of a natural seed trap, so why don't I collect seed bank data? Because seed bank is vegetation minus establishment. It's a great thing, and it should say us more about how the species really disperse. Actually, as you may know, especially with forest species, there is a problem that they don't have large seed banks. So although I was able to find quite a lot of species that were present both in seed bank and vegetation, to get reasonable occurrences or relative frequencies, I found only 8 species. So I took these 8 species, and from their above ground abundances, I made on these abundances I did variation partitioning between environment and space, This was an estimate of dispersal limitation as it has been done in the literature. And then I computed distances, dispersal distances, or the proxies. These were distances from the seed bank to the nearest neighbor in above ground, and also some corrected number of seeds, which was representing observed dispersal. And I took these two measures because dispersal, it has two parts, and one is the fertility and the other is the dispersal distances. And here are some basic results. We can see to what degree the seed bank and above ground vegetation overlaps. These are the black bars. We can see it really depends on a species, that in some species the overlap is quite good, while in other species it's really poor, and there must be some really strong establishment limitation or some dormancy going on. And when I put this pure space and the estimates of dispersal into a graph or into a correlation, what I found was that there was no relationship. It was similar with distances to nearest neighbors and with numbers of seeds, which I don't show here. What we can see is that the species tend to spread over relatively short distances, and they also tended to have relatively low numbers of seeds. And there is one point I had to take into account, and it was the geometrical constraints, because the population is small, and if you have a species that is almost everywhere, then you don't have much possibilities to find a long distance to a nearest neighbor. So when I accounted for this, the species tended to be either dispersal limited or distributed at random with respect to this. And if we go back to the relationship that wasn't really working, I was thinking, why was it like this? So there is one possibility that is pretty obvious, that I have too few species to get anything significant. But the other thing might be that there could be some issues in pattern detection, because these pure spatial or spatial partitions were really low, because maybe the species have some finer patterning, and I had 10-meter lags between plots, so many things may happen in between in this blank space. And the other thing was that I was thinking of this establishment limitation, because I found that it's there, so maybe it's distorting the relationship. So to put this together, I found that there are spatial patterns in the forest understory communities at a small scale. And at the community level, most of these spatial patterns could be ascribed to environment. However, as regarding the pure space, it's hard to conclude about its origin. I was really trying to find a relationship with dispersal, but well, I can't say it's there. It depends how we ask and what we use as evidence. But at least I can say that maybe in some species and under certain sampling arrangements, this pure spatial variation is not a good measure of dispersal limitation. So that's all from me now. Thank you for attention.